I was the first one in 1977, just before I came to the US. I immigrated in 78, and this is the only job I've had since I've been here. I was the first one to describe and publish clinical outbreak of drug-resistant infections caused by drug-resistant strep pneumoniae. Since then, it's spread all over the world. In 2002, we were the second lab in the world to describe the VRSA, vancomycin-resistant Staph aureus, and since then I've been examining the MRSA strains for vancomycin susceptibility, looking at new drugs against Staph aureus susceptibility, resistance patterns, etc. I remember it almost to the day. I was in a lab, it was in Durban, South Africa, which is a city on the Indian Ocean, and we were looking, and luckily for us, not for the patient, but for us, the organism was isolated, the pneumococcus, from the blood, and the second one was from the cerebrospinal fluid. And I don't quite know why we were looking for it, but we put a susceptibility disc on the plate with growth, and there was no inhibition. No quality control, nothing. Anybody that would have looked at that lab would have had absolutely nightmares because it wasn't properly controlled, but resistance was there. But the important thing is that what I've always kept before me in life is what Louis Pasteur said: fortune favors the prepared mind. So Alexander Fleming could easily have thrown out that plate with a fungus that inhibited Staph aureus. He didn't. I could have quite easily thrown out that plate with the pneumococcus that grew right up to the penicillin disc. I didn't. I told the people, test it further. It's as simple as that. Simple as that. In 1995, when the first vancomycin non-susceptible Staph aureus strains came out of Japan, I went down to the clinical lab in Hershey Medical Center, which I'm in charge of, and I told the technologists, I said, folks, from now on, look for vancomycin susceptibility on every single MRSA that you get properly. The technique is not important for this discussion, but properly, and because we looked for it, we found it in a patient in 2002. If we wouldn't have looked for it, we never would have found it. It's that simple, or that complicated. And that's what I try and teach when I give lectures to students. I always tell them, you don't have to be clever to find something. All you need to do is to have an inquiring mind and to look. If you look, you find. If you don't look, you don't find anything. But when people like me are going to disappear, nobody's going to be looking anymore. There are hardly any new drugs, even with letters and numbers, because I get them when there are letters and numbers, when they haven't even got names yet, from companies all over the world to test. And there really isn't an application to the FDA that hasn't got at least one paper from our group testing the organisms in the laboratory, because there's several in vitro methodologies that our group uses and can use that nobody else can use, that nobody else in the world has got expertise to use. I've made sure that my research team are trained in the specialities. One person does technique A, the next person does technique B, the next person does technique C, and they do it all the time. They know what they're doing. It's reproducible, it's trusted, and the work is required. The type of thing we do is just interaction of bacteria and new drugs, how they work on the cell wall, susceptibility of a specific group of organisms with specific susceptibility patterns that I've collected over the years. We have in our freezers a very valuable collection of organisms with very various susceptibility patterns, and then we test those against new drugs. How fast do they kill? Do they select for resistance? What susceptibility is, how they interact with the cell wall, or that's the kind of thing. And we publish basically, I would say, a minimum of 10 papers a year, and I present probably about upwards of 20 papers a year. I like to feel that a paper from the Applebaum group, which is hopefully what they call me, is a gold standard paper. When they see a paper from me, they know it's got the stamp of authenticity and reliability. One of the best compliments I ever got in the world was when we discovered that VRSA, that vancomycin-resistant Staph aureus. I was at a meeting in Europe, and somebody came up to me and he said, you know, I didn't believe this until I saw your name on the paper, and then I believed it. That's what I've developed over these 30 years in the US and 10 years before in South Africa, is a good name. We know, everybody knows, that when I publish a paper, you can take that paper basically to the bank, so to speak. You know that the data there are good, and they're reproducible, and they're reliable, and if you test the same thing, you're going to get the same result. Commercial labs out there that are for profit, some quite good ones, indeed one specific very good one that I'm thinking of, but they make no bones about the fact that they are for profit, and they test tens of thousands of organisms, and they sell them. Then they have a stock and they sell them to people like me when I'm interested, and they make a profit, but they don't delve into the details of the interaction between the drug and the bacteria, which my lab is specialized to do. And you need both. The one without the other is not complete. You do need both. Because of the lack of funding, and the problem is that the type of work I do cannot really be funded by the government, by NIH, because I don't have a null hypothesis. If I'm looking for a new resistance type, I can't tell the National Institute of Health what it's going to be. And they need to know that information in advance before they'll even think of funding anything. So I've got to get my work funded entirely by industry. And because industry is cutting back so severely for so many reasons, nobody is going into the field. I'm basically a dinosaur. When I retire, unless I've trained somebody or one or two of a handful of other people in the world that have, people with my expertise are going to disappear and are not going to be retrained. I am hard put to know why anybody would go into the field that I have. It was a good field 30 years ago because there was still money and interest, but now nobody would go into it. And that's why when I retire, I do not want to let this team go away and lay everybody off, because we're giving a service which is absolutely necessary for the community. But the only way that I can hire somebody and then keep a paternal eye on it when I retire, which I would like to do, is with continued financial support. Unfortunately, everything boils down to dollars. To give you an example, last November, it really looked as though we would have severe problems. And out of the blue, one company came, had 3 or 4 studies, and now we are solvent for the next several months. But it's very, very difficult to work like that. Very difficult.